both the granular cell and periglomerulus cells help in lateral inhibition so that you can sharpen the focus of the smell. Hello, welcome to another episode of Physiology with Shakthi, and in today's episode we're going to do with olfaction. So when we talk about olfaction, let's take an example of a game called The Witcher 3. So in that, Geralt, the protagonist, he talks about his lover, old-time lover, Yennefer, and he compares her smell to that of lilac and gooseberries. So how exactly is this Geralt thinking about this smell and connecting it to that person, connecting it to the memories associated with that person? How exactly is he segregating the smell into different components like lilac and gooseberry, and how does it make it feel whenever he gets that perfume? So that is what we're going to try to understand today. So to start off, just take a look at this diagram. You can see the nasal cavity. So in the nasal cavity, in the roof of the nasal cavity, that is where your olfactory epithelium is present. So there is a epithelial layer which is present, and that takes around 10 centimeter squared area. So this epithelium has the olfactory neuron present. So this is there in the superior part of the nasal cavity, so the roof. So if you know about the nasal cavity, we have superior, middle and inferior turbinates. So normally when we do normal respiration, airflow does not reach a superior concha that well. Only when you take a deep inspiration or you try to sniff things, that is when it maximally goes into your superior turbinate or concha. So that is the importance of sniffing. So when you go sniff something, that is exactly when you are going to be able to sense things better than when you are normally respiring. So that is the importance of the location of that. And in it, there is a epithelial layer, and in that epithelial layer, there are different types of cell. So one cell among that is the olfactory neuron. This olfactory neuron is a bipolar cell. So that helps you initiate the olfaction. So let's take a look at that cell and the epithelium. So this is one bipolar cell. This is a olfactory neuron which is present in the epithelium, and one end of it is thrown into folds. So these are the cilia. So normally there are about 6 to 12 cilia that are present in one olfactory neuron. So this is your olfactory neuron with cilia present. So this is your olfactory epithelium, and you got your cilia which are jutting out. Now there are presence of another group of cells over here. They are called your supporting cells. So these supporting cells, or the sustentacular cells that are present over here, they secrete a binding protein. So an olfactory binding protein is secreted. So you got the olfactory binding protein. Also in this layer, there is a layer of mucus lining over here. So a layer of mucus is there with your olfactory binding protein. Got it? So when there is some substance that need to be smelled, so that substance needs to reach the cilia, where it should activate a set of receptors. So in order to do that, first thing, it should be volatile. So things that are volatile, and this is mucus, things that are lipid soluble, these are better smelled. So you can smell these things better. So volatile things and things that are lipid soluble. So these are the best rate of olfaction. And things generally bind to your olfactory binding protein, and then they start stimulating things over here. Got it? Now, apart from this, in the olfactory epithelium, there is another group of cells. So that is called the basal cells. So these are the basal stem cells also which are present. So these basal stem cells help form your olfactory neurons. So olfactory neurons cannot stay for very long. Especially imagine a case where you're going to smell a lot of pungent smells. So obviously in time they are going to get destroyed. So these neurons can get replenished. So they are formed from your basal stem cells. So this is one area of the nervous system where we know that things can be replaced, neurons can be replaced. So a lot of studies have proved that. So you got the olfactory bipolar neuron. We got the sustentacular supporting cell which secretes the olfactory binding protein. And we got the basal stem cell which helps regenerate the olfactory neuron by mitosis. So by mitosis they help regenerate it. And also this usually stays for around 1 to 2 months, the olfactory neuron. So this is your olfactory epithelium. So consider this one component over here. We'll zoom into this and see what exactly happens over there. So this is what I'm talking about. So this cilia over there, I zoomed it here. And you got receptors which are present in the cilia. So these receptors are G protein coupled receptors. So they are large G protein coupled receptors with a alpha, a beta and a gamma segment. This alpha, is attached to GDP. So this is the receptor which is present. Now, when we talk about these receptors, when we look at the genes, our genome, around 1,000 genes have been discovered which are related to olfaction. And out of those 1,000 genes, around 400 of those genes are responsible for receptors of olfaction. Now, the different types of smell are there. So to segregate them, there are different types of receptors. We know around 400 types of different receptors which are genetically encoded. And then further differentiation can still be done. So we'll come to that a little later. So for that work that was done, Linda Buck and Axel Rod, they got the Nobel Prize for discovering this. And so once we get the smell, it stimulates that receptor, which is the G protein coupled receptor. G alpha stimulatory is activated, adenyl cyclase is activated, cyclic AMP is activated. So finally the cyclic AMP is going to open up a channel. So this channel is permeable to sodium, calcium and potassium. And out of that, the major role is played by your calcium. So calcium influx here would result in not the action potential, a graded receptor potential. So right now there is a receptor potential which is generated. It has not gone into action potential yet. Now because of the calcium, a calcium induced opening of chloride channels are there. Now these cells, these neurons, they have high intracellular chloride. So chloride is a negatively charged ion. So it is high intracellular concentration is also there in this neuron. So what does that mean? High concentration to low concentration. So chloride is going to go out. And because chloride goes out, inside becomes more positive. So now because of the influx of calcium plus calcium induced opening of chloride channel, there is the graded receptor potential going into action potential, spike potential. And that is where your first signal of olfaction starts. So you get the picture? There is a volatile substance or any odorant, and that binds to odorant binding protein, and finally it stimulates your G protein coupled receptor. G alpha is activated, adenyl cyclase is activated, cyclic AMP is activated, cyclic AMP phosphorylates and open up a channel that is permeable to sodium, calcium and potassium. More so to calcium. Calcium influx will result in a graded receptor potential, and that will result in opening up of a chloride, and that chloride channel means chloride is going to go out because of high intracellular calcium of chloride, and that ultimately will lead to formation of the action potential. So that action potential has been formed, which is now going to get transmitted through the olfactory neuron. So that is about your epithelium, its receptor, and how the transmission starts. So that is the first step. Now next thing, we need to understand what happens from here on out. So if you done anatomy and if you've seen the base of the skull, you would have noticed the cribriform plate, which has a lot of pores in it. So through these pores, these olfactory neurons are going to come out through these pores. So we have your olfactory neurons, we have the cribriform plate with pores present in them. And let's just draw another olfactory neuron over here. So just know that this olfactory neuron is kind of sensing a different smell because it has different receptor compared to this olfactory neuron. Okay, now this is going to pass through your cribriform plate and go over here. At the same time, some other olfactory neuron which senses a similar smell is also going to come along with it. Will this come over here? No, because that is a completely different type of smell. So this is also going to go separately over here. And consider something similar to this smell is also coming from somewhere else, and that goes along with this. So the first set of segregation after your receptors taking place over here. So I had initially mentioned how there are different types of receptors, 400 different types of receptors are genetically known. And further on, they are going to go and combine separately at a different location. So that location is what is called the glomerulus. So there is something called a glomerulus over here. And this side, there is another set of glomerulus. So what is this glomerulus? So for that, just come back to that earlier diagram I had shown you about the nasal cavity. So if you take a look at the olfactory bulb, you can see that this is actually one of the most primitive of the cranial nerves. It directly juts out from your brain. It's actually part of your brain which kind of juts out from the forebrain. You see that? So that forms the olfactory bulb. So this is your olfactory bulb which is present over here, which is kind of a direct extension of your brain. So this olfactory bulb is extending, and in the olfactory bulb there are group of cells. So one group of cells is the mitral cell, and over here also there's a mitral cell, and another group is your tufted cell. So we got the mitral cell, we got the tufted cell, and they have their dendrites which are passing through. So can you appreciate that? This is a tufted cell, this is a mitral cell, it has its set of dendrites, and this is the axon emergent from it. So this glomus is a conglomeration of many such dendrites of mitral and tufted cells. So around 25 tufted and mitral cells are present in the glomerulus, and this conglomeration of the dendrites which are connecting each other over there of the axon of olfactory neuron along with the dendrites of the mitral and tufted cells. So that together forms a glomus. Understood the picture what a glomus is? Yes, and also we've already seen how separate glomus are there for separate sets of receptor activation. So similar type of smells are put into one glomus. Is that clear? Yes. So next thing is to see what happens from the glomus, and from the glomus the transmission should go further. But before that, just like your visual pathway, we have amacrine horizontal cells which need to sharpen your focus. So when one activation of this happens, it should not keep spreading and activate everything else. You lose focus. Instead, you'll smell something very vague. So to stop that, there are set of neurons which are present over here. So this is one glomus, this is another glomus. So periglomerula. There is one cell over here, which is a periglomerula neuron. So these are periglomerula neurons. So they help in inhibition, lateral inhibition, and sharpens it. So make sure that anything that stimulates this does not spread and is limited over here. Understood? Also, between mitral and tufted cell, you can see the dendrites, how close they are. So we need to make sure that stimulation of one particular thing does not end up spreading throughout and stimulating each other. So that is why we have another set of neurons, which are the granule cell. It goes here, and again helps in inhibition, lateral inhibition. So both the granule cell and periglomerular cells help in lateral inhibition so that you can sharpen the focus of the smell. Now, one thing about the granule cell is that it does not have an axon. It does not have an axon and only has dendrites. So this is a structure, a neuron, which has only dendrites without an axon. So how does it help in transmitting? That is an example of dendrodendritic. So dendrite, a dendrodendritic transmission. Otherwise, normally this is an axon and this is a dendrite. So axodendritic transmission and axosomatic transmission are the most common type of synaptic transmission that we know of. So this is one example of dendrodendritic transmission, which is the granule cell which are present in the olfactory bulb. So am I with you till now? So we got the periglomerular cells and granule cells, which through lateral inhibition is going to sharpen the focus. We got the glomus, which has a conglomeration of the dendrites of mitral and tufted cell synapsing with the axons of or the olfactory epithelium. And each set of neurons of a particular type of smell is going to go to separate glomus like this. And we got around 400 different types of receptors which are present. And also you need to know that around 25,000 of neurons converge onto 25 different types of glomus. Okay, so after this, what happens? So we got the smell, signal transduction has happened, it has gone over here and it has sent its impulses to your mitral and tufted cell, and then it is going to go further. So what else is going to happen now? How is your brain realizing what type of smell it is? So that is why you need to understand two types of striae that is going on from here. A medial striae and a lateral striae. Two sets of pathways are diverging from here. One is a medial striae and a lateral striae. The medial most striae is a little more primitive. Lower animals and all that, you can see higher medial striae functions. And in more evolved animals, the lateral striae has also come into play. The paths that go to the medial striae are responsible for very primitive reflexes, like how animal, when you give a food, keep a food with an odor close to it, they exhibit behaviors like lip smacking and all that, like that, yeah, and they start salivating unintentionally. So all these primitive things that happen, these reflexes that happen, which are related to olfaction, are going through the medial striae, while the lateral striae is a comparatively new part which goes to parts of the cortex and helps in actually differentiating the different types of smell. So that is the lateral striae.